Hello, I'm Andrew. I share on social media for UAF, and I am here at the Wood Center where URSA's Research and Creative Activity Day poster session is just about to, it's starting to happen. Oh, there's someone. Let's go talk to this person. He doesn't know that we're Facebook Living. We're going to come talk to him live on Facebook, but here we are. Are you ready?
Ready.
Hello, what is your name, please?
I am Paul Lair, and I am the Vice President for Academics and Student Affairs at the University of Alaska.
And I see you looking at this amazing research poster right here.
Right, this is a poster from the School of Education, and it's looking at how young children perceive their world and their environment through their drawings in terms of what they're doing for recreation and subsistence activities.
Do you get to award some poster awards? Like, is this a front runner?
No, I'm just here because I like to see what's going on with our students here at the universities.
You've seen lots of research posters in your career. What makes a good research poster? What do you? If I went to look for a poster, what should I look for?
You have to talk to the students who are doing it, because you have to look at their enthusiasm and how they really got into the research and were really embracing it. I think that's really the most exciting part about any poster is the researcher's engagement with their project and really understanding it and being excited about it.
And posters are a time when the introverted scientists who are usually head down in data come up for air to share their work with people.
Right, and they have to really, and that's the challenge. I mean, it's easy to work in a lab and to do your research, but to explain it to somebody else who's not in your field is something that's really more difficult for a lot of students to do and a lot of researchers to do, especially in somebody who's not in the field. So I'm not an educator, but can I understand what this student is doing through this poster is a really important part of what we're doing.
You've probably made posters yourself.
Yep.
What's the hardest part about making the poster?
Trying to cram too much information into a small space and realizing you can't tell the whole story on that poster, but distilling it down to those key elements is really the hardest thing about making a poster and then being able to explain that even when you're not there. And that's the other side of making a good poster is, can somebody explain it even if I'm not around to explain it to them?
Is there a risk going the other way? Too many pictures, too much fun, not enough info?
Yes, that's true as well. And you really ought to be able to understand what the purpose of the research was, and not just simply the pictures or the graphs or kind of the more speculative kinds of things. But what really was the research? How did you evaluate your data? That's a very important part to convey in a poster.
Is this almost more scary than a final exam? Putting your stuff together, presenting a poster, and putting yourself out there for people to talk to?
Absolutely. I think it's more rewarding because you get a lot more feedback. When you're just doing an exam, you get a grade and that's it. But this one, you're out there, you're explaining it, you're getting feedback, you're looking for ways to do more on that research. And I think it is harder in a lot of ways because you are putting yourself out there. It's very personal. It's much more personal than an exam.
Thank you very much. I'll let you keep on perusing around. I'll go check out some other posters.
All right, thank you.
Thanks. All right, let's see who we can find. Maybe there's a researcher around we can talk to at their poster, because as they say, they've got to stick by their posters so they can talk all about it and give the interpretive information. Oh, there may be someone at their poster right now. Are you at your poster? Can I talk to you live on Facebook? Excellent. All right, here we go. Have you had anybody talk to you yet? Is this your first one? All right, let's turn this around.
Okay.
All right, what is your name?
My name's Allison Burke.
Allison Burke, and what are you studying?
I'm studying Antarctic fish.
Antarctic fish. Did you get to go to Antarctica? That's my first question.
I did not. Only the grad student and the professor got to go. I'm just the laboratory tech.
Okay, so here's your poster, and what is your poster title?
Sequencing hypoxia responsive genes in Antarctic fish.
That sounds pretty interesting. So, hypoxia, is that when you don't have enough oxygen?
That's exactly what it is.
Whew. Okay, so what is tell me about your poster.
All right, there's these 3 species, and this guy is the special one because he doesn't have a hemoglobin, which makes it really hard for him to get enough oxygen around his body, especially in hypoxia. And our big concern is that as the oceans warm due to climate change, warmer water doesn't hold as much oxygen, so these fish could be in danger of being threatened or going extinct. So we're looking at this protein called hypoxia-inducible factor 1. And there's this pathway shown here where under normoxia, generally it'll break down because it doesn't get hydroxylated, except it's a weird thing in fish. They're pretty sure that it doesn't actually break down. and that's kind of a very unique thing in animals. But it does the same under hypoxia, pretty much. It binds to another subunit, and then binds to the DNA, and it starts a whole bunch of transcription and makes a whole bunch of genes work.
Wow.
Yeah, and the weird thing is, in this hypoxia, there's an insert of glutamine and glutamic acid amino acids that they found in the alpha subunit. Here you can see each of these is the number of amino acids that are inserted. You can see in the red-blooded, it's a few, and then if you get down to the ice fish without hemoglobin, it's this huge 34 amino acid insert.
That's a lot of gene editing.
It is, yeah. So the problem with that is we don't know if it inhibits the activity of HIF1 yet. So that's kind of what I was testing. But in order to do that, we need to figure out how to quantify its activity. So we found these 3 genes, which are 3 of the genes of, I think it's thousands that it triggers. And I made some degenerate primers and replicated them and cloned them.
Look at those gene sequences right there. You cloned them. You did some cloning?
Yeah, I put them in plasmids and E. coli cells and grew them in plates.
Whoa! No, there was always only good cloning. No bad fish clones came out of it.
No, it was only for little plasmids, not for whole fish.
Oh, okay. I've seen enough movies to know that's probably not a good idea. Oh, yeah. What did you find? What were some of your results?
So far, all we've gotten is a couple of sequences. So these are the different species. There's an Ocerotus, a Corioceps, and a Gibberifrons. And they're all aligned here. You can see little differences in each of them that are highlighted. So what we're going to try to do is create primers that will sit down on a section like here where they're exactly alike, and then it'll be able to replicate the entire thing in all the species. And once we do that, then we're going to be able to do quantitative PCR, where we find out how much the gene is actually transcribed. And if that's higher under hypoxia than normoxia, that means that HIF1 probably still works.
So ultimately, it's how these fish can survive a changing ocean.
Yeah.
Because if the fish go, then the next step are birds and higher mammals, and then humans.
Yeah.
So it's really the future of humanity that you're studying.
Really, it is. It's very important.
What's it like to put a poster together? When people come to the poster session today, what should they be looking for a good poster versus a bad poster?
I've always been told you got to make it big and you got to make it colorful. Because when people come in, they're probably not going to read all these paragraphs. This is the stuff they're going to look at. They're going to look at these pictures. They're going to look at the fish. Sometimes they'll look at the citations, but
Like, is my paper in there? Did
Yeah.
they look at? Did they put me in there? So you want big, colorful, and then you get to fill in all of the blanks.
Yeah.
Are you nervous? You did a great job. You did a great job
you.
just now. Are you nervous about the day? Is it hard to get out of your lab and get out into public?
I'm a little nervous. I gotta rely on my charisma.
Well, you did a great job speaking with me. Thank you so much.
Thank you.
And are you here all day? Did you just take a break?
I have some classes that I go to, but in between classes I'll be
You'll
here.
be right here. All right, so people can find you here. Thanks a lot.
Thank you.
Thanks for help saving humanity.
I try.
So they're down here in the Wood Center. Might wander around, see if anything else pops up. That was great. That was just what getting a poster is like. Not all the researchers are here yet. The poster's all up. They're probably getting coffeeed up before they come back out here for the afternoon. But there's, oh, look, over here I see someone using virtual reality. Let's go over, and that always looks good on Facebook. All right. Here's someone in VR right now. I'm going to turn it around. All right, you're live on Facebook. Get ready.
All right. We prefer not to be live on Facebook.
Yeah. Oh, okay. Well, then I won't put you live on Facebook. But you look so cool in your virtual reality gear.
We're still setting up.
Oh, they're still setting up. Oh, okay. So you can come over. Can anybody use the VR stuff if they come over here later today? I'm just showing me. I'm not showing you guys right now. So you can come. You can use some VR, and you can visualize snow depth. Can you dive down into the snow yourself? That's exciting. Okay, we'll see. So there's VR down here. There's lots of posters. There's some unmanned aerial vehicles going on here. There's some more explanations over here. Ooh, that looks fun. I'm just going to turn on. How are you guys?
How's it going?
Good. All right, that's it. Yes, live streaming on Facebook.
Well, yeah, we're counting microns right now.
You're counting microns right now?
Yeah.
Those are so small.
Yep.
Like, if I was to hold, is a micron like that big? How can you even?
1/1000th of an inch.
That's It's so tiny. How do you count it? What's counting it?
A laser inside of there.
Lasers and microns?
Yep. Yep.
I don't know what is even going on. That's so cool. What are you counting it for?
We're simulating air environment within the fuselage of a commercial aircraft to help reduce disease transmission between passengers.
Oh, nice. So
you don't get sick on the plane.
you're saving people from getting sick while they travel.
Yep.
Thank you. I appreciate that. All right. So that's all sorts of cool stuff. Thanks, you guys. How many microns you got so far?
We've had this on for half an hour, and we're at 100,000.
100,000 microns. Is that a lot?
Yeah, there's 100,000 particles that could potentially be bacteria or virus.
So wear a mask when you come to the poster session? I don't know. Thank you. Oh, wait. Someone was just, oh, look, there's someone I know. Okay. Come down and check it out down here. They're down here till 4 o'clock today. Bunch of research stuff to find out. Are you presenting a poster? Oh, look, one more person. Are you going to go live on Facebook?
Sure.
Oh, great. Okay, here we go. Let's talk to this person.
Kelly.
Yes, okay, now it's facing you.
Okay, great. Hi, I'm presenting a poster on when I went to New York for the URSA's University Resident Theater Association, and I got to perform 2 contrasting monologues in front of at least 30 schools. So, yeah, so I'm presenting a poster on that and what the URSA is. There's my acting headshot, my resume.
There you are. And your resume. Yeah. Nice. Oh, you just added a new show that you were in. So usually people think scientific posters are like counting the microns, or a person I was talking about the fish genetics over there, but this is more of a liberal arts research that you do.
Yeah, I mean, and it's more of like a conference type thing that I went to. Like over here we have people who actually have other liberal arts types of stuff, you know, not just like physics or biology. They have probably like more of like the social science type stuff. So it's not just about like research on like microns, like you said.
Right.
It could be like social interactions with people and other stuff like that.
So I think that your research is acting. Your research is going and putting yourself out there. Then your research happens in the theater itself. Mm-hmm. In fact, they call the lab theater over there for that sort of stuff. Are you here all day? You're going to talk about?
Yes.
Look at all these people that saw you. All those people saw you. Is it more difficult to be on stage for a couple of hours, or standing in front of your poster talking about it for several hours?
I think just talking about it in front of my poster is a little more difficult.
Well, thanks for taking some time to share and to widen the scope of what we're talking about here and the research happening at UAF. Thanks, Kelly.
Thank you.
Okay, great. I'm going to go so that I can get over to work myself, or I can do this live all day over here. You can see the full breadth of research that happens here in Fairbanks. It's not just in a lab counting up fish genetics or cloning fish or studying microns for the atmosphere. It's also the quality of life and the arts that happen in the world. All of that sort of research happens on the Fairbanks campus, and they're presenting it all today down here. How long are you guys here today? Till 4?
Till 4 o'clock.
Till 4 o'clock. All right, I'm going to turn you around. We'll sign off with you. Are you ready to go live on Facebook? Okay. Till 4 o'clock.
At 4 o'clock, we're having our award ceremony for Dean's Choice Awards, and there's going to be food that's free. Come down.
Free food and prizes.
Can't go wrong.
All right. Thanks a lot.